Good afternoon, everyone. Well, welcome. We're starting our new year of the Alfsian Family Center for the Prevention and Treatment of Parkinson's Disease lecture series. I think many of you know that we have these lecture series once a month, Thursday, 2 o'clock. We try to keep it at this time as much as we can. And we've been very fortunate. I think probably one of the advantages in adopting Zoom in our normal repertoire of days is that we could bring guests from abroad, like our guest today, with a minimal expense, a lot less flying time, and gives us really the opportunity, without booking very far in advance, to really get hot topics as quickly and as easily as possible. And that's going to happen today. Just since it is our first of a series, and some of you may be new to the center, I do want to tell you a little bit about it. So our mission at this center is to find treatments, cures, understanding mechanisms for Parkinson's disease. And we take this mission very seriously. And that's why primarily our work is between laboratories on campus at Tel Aviv University and laboratories in the hospitals that are affiliated with Tel Aviv University. Now, the center itself is co-directed by Professor Nir Giladi, who is here with us today, and by myself. And so this isn't my field, but I've learned an enormous amount of Parkinson's and enjoyed it a great deal. And for those of you who know Professor Giladi, of course, he's one of the world's experts in the area, and it's really a privilege to be able to work together. We also have an absolutely wonderful team who I'll introduce to you. Rinat Zalzowski, who just raised her hand, who helps organize these lecture series and almost all of our other activities in the center. We have Amit Eshel, raise your hand, Amit, who also is very busy, deals a lot with the finances and the running of the center. And it's really been, I think we're now going on 2 years that the center has been going on at Tel Aviv University. I think that we've made, and we as in the royal we, I haven't done any of the experiments myself, tremendous progress. We have grants, competitive grants that we give out, $25,000 and $100,000. We have scholarships for master's, PhD, and postdocs. We have travel scholarships for the days that people used to travel to conferences. Hopefully that will be happening again soon. We have these lecture series. We have an annual event, a very large one, not only for scientists and clinicians, but also for patients. And we have a number of other smatterings of different types of meetings throughout the year. And the goal is to learn as much as we can and to make the necessary leaps that we have to. We're extremely fortunate that we had a very generous donation by the Alfsian family. It was started by Alan Alfsian, who's very dear to us. He passed away about 1.5 years ago. But we're very fortunate that his 4 children and grandchildren, so it's really 3 generations, have continued giving and taking a very active part in our center. So now let me tell you about our guest. So I know Sebastian for many years, actually interviewed in Israel in 2007. He reminded me of that. But before then, he did his first degree and his PhD at the University of Buenos Aires in Argentina. And then he took a faculty position at the Hebrew University in Jerusalem. And a couple of years ago, you can remind me how many years ago that was, Sebastian joined the faculty at Brandeis University. Now, we'll hear about his work today that's connected to Parkinson's disease, and that was the interest in our inviting him. But his research really focuses on molecular neurobiology and RNA metabolism. And he's done really groundbreaking work in the area of circular RNAs. And today we'll hear how he's fusing that world of his and looking at Parkinson's. So Sebastian, thank you for waking up so early and joining us. We're really thrilled to have you and to spend at least this hour together. And I hope that we can do it in person sometime soon.
Okay, thank you very much for the introduction. Thank you very much for the invitation, Karen. I'm very happy to be there, at least virtually. And I will share my screen. Yes. Okay. Okay. And again, thank you very much. And I'm very excited to give this talk. So this is the first time I'm going to give this particular talk. We have been interested in Parkinson's disease for a long time, and I'm going to try to do today, is to link some of the molecular work we do in the lab with this study that we carry out in the last few years together with the lab of Mona Sorek at the Hebrew University. And this study we started in the Hebrew University and continue collaborating when I came here to Brandeis in 2016. Okay. So the title of my talk is the role of circular RNAs in aging, Parkinson's disease, and other neurodegenerative disorders. And I want to start by this phrase, which I think symbolizes our journey working on circular RNAs, and I think the scientific journey for many people. And it is that says that all through passes through 3 stages. First is ridiculized, second is violently opposed, and third is accepted as being self-evident. And this happened to us in particular when working on circular RNA, which has been very gratifying in the long run, but very hard when we started working on this topic in 2013. So I'm going to tell you a little what circular RNAs are. Circular RNAs have been known for a long time. Indeed, the first paper mentioning them was an electron microscopy from 1979, showing these circles of RNAs in HeLa cells. And what the researchers noticed back then is that these molecules, these circles, were resistant to proteinase treatment. This means they were covalently close nucleic acid molecules, and they were only degraded by endonucleases, but not by exonucleases. So what these circular RNAs are from, or where they come from? As you know, mRNAs, or most mRNAs in mammalian cells, or generally in eukaryotes, have exons and introns. The introns are spliced out from the message, which also has a 5' cap and a 3' poly A tail. So circular RNAs come also from protein coding genes, but are generated from the messenger, from the mRNA or the pre-mRNA by a process called back splicing, in which a 5' splice site, instead of splicing to the next 3' splice in the intron, is going to splice backwards, going 1, 2, 3, or 4 exons upstream. And this will generate a circular molecule which has no free 3' or 5' ends. So this molecule is going to be very long-lived because it has no, the normal pathway for degradation of mRNA are not going to be valid for this molecule because there is no 3' and no cap. So they are very long-lived, very stable. So what are circRNAs? So circRNAs contain 1 or more exons. As I told you, most of them are between 2 and 3 in mammals, but you can have up to 5 or 6 exons in a circular RNA. The size of a circRNA used to be between 250 and 1 kilobase long. They are generated by exact, they are compressed within exact splice junctions. This means they start and end with an exon or within exons. And this is because the spliceosome, or the same machinery that makes the splicing, is going to make these circles. CircRNAs are cytoplasmic. So they are produced in the nucleus by splicing, and then they are exported by a specific pathway to the cytoplasm. They are very abundant, and they have been under study basically because you need to look for them to find them. Because I will show you the slide in a second, but they have a very particular sequence that allow you to differentiate from the linear RNA. But again, you need to look for it. Otherwise, you're not going to see it, and that's why probably they were under the radar for so many years. And another important thing is that they are evolutionarily conserved. In particular, genes that make circRNAs are going to probably make the same circRNA or a very similar circRNA in another species. And I'm not talking just about sequence conservation, because this is obvious, because many of them come from the same open reading frame. They're encoding sequence. The sequence is going to be conserved, but the fact that a given gene is going to give make a circle in a given organ, it is conserved through evolution. So circRNAs can be identified de novo by RNA sequencing. When you perform an RNA sequencing experiment, you usually get the RNA sequencing reads. You align them to the transcriptome, and a lot of them are going to just fall within exons, and then they are very easy to assign to a given gene. some of them are going to be generally, when you have an mRNA, to the junction. So they're going to be a piece of the RNA secretary in one exon and a piece in the next exon. And this, because you align to the transcriptome, you're also being able to assign to an mRNA. Now, for assigning to a circular RNA, you are going to look for a RNA secretary that starts in one exon, but finishes in an exon upstream and in the opposite direction. So you really need to look for it. And these particular reads are the only ones which are specific for a circular RNA. So maybe this is important. It's clear. If you have RNA6 inside this exon, you cannot know whether they come from the circle or they come from the linear, but you need those split reads that go upside down to be sure that they come from a circular RNA. CircRNAs have a few properties that make possible to distinguish from the linear RNA, not only the presence of this particular junction, but also the fact that they are poly A minus, they don't have a poly A tail, and they are resistant to exon nucleases. So this is just a northern blot. And here we have these are the two different circular RNAs. If you, this is just a northern blot for the gene plex A, that of course makes a linear RNA and a circular RNA. And if you look in total RNA, just you extract RNA from, in this case, this circRNA is from flies. So extract RNA from fly brains, you run it in the northern blot, you're going to see two bands, and you use a probe against the exon that makes the circle. You see the band of the circle, you see the band of the linear RNA. Now, if you take only poly A plus RNA, you're going to see only the mRNA. And yes, sorry, this is over this head, okay. And then the same with other circRNAs, even if they have several bands. Now, again, circRNAs have no 3' end. So if you treat the total RNA with an RNase R, which is a 3' exonuclease, you're going to degrade the mRNA, but not the circle. The circle is stay there. So this is a very useful way to verify the circularity of the molecules. So in this case, you can see you have a mRNA running a northern blot, this circle, this mRNA, and the mRNA disappears with the exonuclease, but the circle no. So this is another property, and also illustrates how stable these molecules are in cells. So when we start working on this, there were many open and exciting questions. One, how are the circRNAs generated? Where are they expressed? And what do they do? And not only our work, but the work of several labs, and I would say now several dozens of labs around the world have helped us understand some of these properties. So circRNAs are going to be generated not from any exon. They're going to generally be made from exons of which are constitutive exons. This means these are exons that are used to be in all the messenger RNAs of a given gene. So every time you make a circular RNA, you make it at the expense of the linear RNA. So we showed a few years ago that the circRNA, by only virtue of being done, already has a regulatory potential because decreases the amount of the mRNA from the locus. And I need to say that a lot of the genes that host circular RNAs are very important genes in the brain for synaptic plasticity, receptors, etc., etc. So this has already by itself, even if the circle doesn't do anything else, this has an important regulatory role on the amount of mRNA generated from a locus. On the other hand, we also show that the information for making a circRNA is in the introns flanking the circle, and circRNAs usually are generated when this splicing of the intron, which is upstream and downstream of the circle, is not very efficiently spliced and tend to interact with each other either by complementarity between the two introns or by proteins that are going to bind to the two introns and bring them together to make the circle. So that's the way the circles are being done. There is a lot of work now on the splicing factors, specifically brain-specific splicing factors, which are important for making circles. So where the circRNAs are expressed, that's another question we were interested in. And we show, I mean, there's now a lot of study, but we show in 2015, together with the Rajewski lab, that circRNAs are strongly enriched in the brain. So if you just do RNA-seq, and this is the same for starting from flies to worm, to mouse, to rat, to human, you have around one more order of magnitude of circular RNAs in the brain than in any other tissue. here you can see it. Muscle also has a fair amount of circular RNAs, but other tissues in the body have much less. So brain is the main tissue when talking about circular RNA, and the circular RNAs are encoded by neuron-specific genes. We also saw that found that circular RNAs are upregulated during neuronal differentiation in human and mouse, and they show up in around the time that synapses are formed. So if we do a primary culture in date, I think it's day 22, they have a big spike in the formation of the circular RNA. And it's something also that what you see during development, brain development, in all these systems is basically the same. Also, we found that there's a large number of circular RNAs which accumulate in synapses and which degrade upon neuronal activity. I mean, the degradation, it has been not done by us, but in the other study, but there are now several more studies showing that neuronal activity modulate the levels of circular RNAs on the synapses. So when I talk about circular RNAs being abundant, I'm talking about the following. So I'm talking, if you have a given mRNA, for example, serotonin receptor, that you will believe this gene, the main product of this gene is to make an mRNA that encodes the serotonin receptor that also makes a circular RNA. And what happens if you measure the amount of the circular RNA, actually the amount of circular RNA produced from that locus is more than the mRNA. And this is not one or two genes, but you can see that especially in humans, you have thousands of mRNAs that are more abundant. The circular RNA is more abundant than the linear RNA. So we are talking about thousands of genes that the main product, at least in terms of quantity of RNA, is a circular RNA rather than an mRNA. And I'm talking about genes that I'm sure you have, you know in the context of neuronal differentiation, neuronal function, adhesion molecules in the brain, etc. So they are very important genes for the brain, which the main product is actually a circular RNA. I don't say the most important one, I say the most abundant one. We don't know whether it's the most important one. So the circular RNA have very specific patterns of expression in the brain. And this is published, so you can just go to the paper and see it. So some circular RNAs are expressed in more regions, in specific regions in the brain than others, more specific or less specific. And what is interesting is that the pattern of expression of the circular RNA not necessarily follows the pattern of expression of the linear RNA. So it looks like, for example, some genes like RIMS2, for example, the circular RNA is mainly expressed in the cerebellum, while the mRNA is expressed among different places in the brain. Of course, this could be due to the stability of the circular RNA, but the synthesis of circular RNA being more specific in one part of the brain than other is actually a key player here too. Another interesting feature of circular RNA is that the circular RNAs accumulate with age in the brain. And this has been observed in Drosophila, in C. elegans, and in mouse. And this, again, can be due to the stability of the circular RNA. And I think it has an important role in aging, as I'm going to show you later on. So maybe the most, I try to give a little of a longer background just to make you aware of how excited we were to work on these molecules in the context of brain function and in particular neurodegeneration. Because again, very abundant in the brain, again, accumulate with aging, and some of the functions of the circular RNAs could be linked to disease too. So we showed a few years ago that this particular circular RNA encoded for a protein involved in myotonic dystrophy, which has as a role to control the levels of the protein. So the protein is a splicing factor, and the idea is that when the splicing factor is very high, the splicing factor go back to the nucleus and deviate transcription from its own production to the production of the circle, and then also bind to the circle and localize it to specific places in the cell. There is another work that shows that the particular circular RNA is very important for neuronal differentiation. And there is another circular RNA, CDR1 antisense, that has, at least when knockout, some drastic effects on behavior and on postsynaptic currents. So regarding how the circRNAs achieve those functions in physiology in the brain, so we don't know a lot about this, but there is a lot of hypotheses about it. So for example, this circularRNA called CDR1 antisense binds to a microRNA, has 71 binding sites for the microRNA miR7, and regulates the amount of miR7 and the activity of miR7, which is important for early gene expression following activation, neuronal activation. We also show, and I promise this is one of the last background slides, that some circRNAs can produce proteins. So they can be template for protein translation. It's not just that they will make any protein. So the circRNAs share the start with the coding gene. So if you have if you're working in a circularRNA, which is generated from the serotonin receptor, the peptide generated from this circRNA is going to share the N terminus with this protein, which could potentially act as a dominant negative or a regulator of the translation of this gene, for example. Not a lot of not all the circRNAs are translated. We don't even know how many, maybe 5%, maybe 10%. They are also translated by membrane-bound ribosomes, which is also very interesting because many of them come from hosting genes, which are membrane proteins, and it's not clear where the peptides are afterwards from the circles, but they are translated for this particular pool of ribosomes. And we found a few circRNAs that the protein is highly enriched on the synaptosomes. I mean, since our paper in 2017, they have been, I mean, there was a few more papers in 17, but nowadays, like, I would say a few hundred of papers showing circRNA translation in different parts of the body and the peptides, some of them having functions. Okay, and the translation is cap independent and induced by pathways that induce cap independent translation. So there is a lot of ideas of what the circRNAs could be doing. A lot of speculation is on transport, as circRNAs are signaling molecules. SignalRNAs are devices for translation of protein in synapses, or maybe to deliver cargos on synapses on distal places on the cell. So many circuits have been found to be associated with microtubules also, but again, there is very few papers on actual molecular functions of circRNA. Most of the papers focus on physiological function. CircRNAs are also relevant for neurodegenerative diseases. And what we knew when we started this project in 2016 was that certain circRNAs accumulate with age in the brain. Many circRNAs are hosted in disease-associated genes. For example, there is circRNAs which are within genes responsible for Alzheimer's disease. And there is at least one of them that is strongly misregulated in AD. And this is an old paper. There is even more also in the animal models of AD. Several circRNAs are misregulated. And also, one of the key factors involving ALS foods, it is actually a circRNA producing a protein. So it's a protein that regulates the production of many circRNAs. So back then when we started working on this project, there was some link between circRNA aging and neurodegeneration. Now there is even more. And I think our work proved even more. Okay. So our question when we start working on this is, how do circRNAs level change in PD brains? And can we use this to biomark Parkinson's disease? And for this, we collaborate with the lab of Mona Sorek, and we were supported by the MJ Fox Foundation for several years. So what we did is we took samples, we asked the Netherlands Bank to send us brain samples, post-mortem brain samples from amygdala, substantia nigra, and MTG. This is the number of samples that we got initially for doing our study. And we basically did purify RNA from these samples, verify the quality, make RNA seq libraries. And these are not normal libraries because they need to have, we want to look at the circRNA, so they need to have, they're not poly A plus RNA. So we basically deplete the ribosomal RNA, and from this ribosomal depleted sample, we do RNA seq total library. We have more than a billion reads from these samples. We have like 5-6 billion reads now, I think this is not even the complete number of reads. So we have a lot of data, a lot of circRNAs detected on the samples. And we found many genes which are differentially expressed in the substancia negra of the Parkinson disease, which makes sense. And this is nothing new. Among them, of course, tyrosine hydroxylase is severely diminished because you have much less dopaminergic neurons there. And we have the pathway that we found alter in these samples are related to normal pathways involving Parkinson. Here is just dedoublating what things are upregulated or downregulated. Although having described before, we also found that many RNA proteins involved in RNA metabolism are misregulated in the substancia nigra of the individual with PD. So then we wanted to try to see what happened with circular RNAs. And the first thing we just did is try to see whether using the circRNA data, we can differentiate between Parkinson's disease and control individuals. And this is actually, this slide. It's actually not the case when we try to do just clustering based on the circRNA data, this cluster by brain region. And this also brought to our attention that the circRNA pattern of expression is very specific for the different brain region. As you can see, there are more genes which are specific to the region, that more circRNAs specific to the region than the circRNAs which are shared between the regions. And there was also some circRNAs that we observed only in the PD or in the control individual, but most of the circRNAs we found in all the individuals. When I was talking here about many circRNAs being specific for the brain region, this was interesting because you could ask me, maybe this will happen the same thing when you look at the mRNAs, but actually it's not the case. So mRNAs tend to be shared between all the brain regions, and with few mRNAs being specific for one or two of the three regions. But the circRNAs actually seem to have more specificity than the linear RNA, and maybe more useful for clustering the samples. And this has been described before in our paper in 2015. So, but it was nice to see it now with the with the smaller regions, and also in the human brain, because what we saw before was mainly in mouse. So when we look at the number of circRNAs in control and Parkinson disease individual, what we expect. So circRNAs are strongly enriched in neurons. So what we were expecting is that in substancia nigra, you have less neurons, you're going to have less circular RNA, and this is what we saw actually. But for our surprise, we also saw that we have more circRNAs in the other brain region, and this effect was even stronger than the decrease on the substancia nigra. And to be honest, we don't have an answer why this is, because we try to understand from whether we see something like this from the mRNA point of view for those genes hosting the circles or any other phenomena, but we could not make sense of this. And if someone has an idea, we will be grateful. So now there have been some links between circRNA and production and RNA editing, but nothing very clear. But what we found is actually that there is an anti-correlation between the levels of the circRNAs in the MTG and the AMG, and having that the regions that have more circRNAs have actually lower editing levels. So maybe the increase on circRNA is driven by this less RNA editing level, which have not been described in other regions, in those regions before in individuals with PD. Have been described globally in individuals with PD, but not in this region. And yeah, we actually have a correlation, but again, we don't understand what exactly this means, but we feel there is something here. One question that we have is whether the circRNAs accumulate with age in the human brain. This has not been done before, and because we have so many samples from different regions in the brain, we could just ask a very simple question, whether the amount of circRNA correlates, for example, with the age of the individual, whether it correlates with the sex of the individual or with any other parameter that we have in these samples. The main thing that we found is that the circRNAs correlate with age. The correlation is quite good in the substantia nigra, but not in the other brain region. So if you take healthy, these are the healthy control patients, no PD patients, and you can see that as the age increases, the number of circRNAs, total number of circRNAs increases. Now, interestingly, this correlation was completely gone in the individuals with the PD. It's again just a correlation, but it is we were excited to see these two things. And again, this was the first time someone described age-dependent accumulation of circRNAs in the brain in humans. So we didn't find a lot of circRNAs which are differential expressed, because that was our initial idea, to try to find circRNAs which are differential expressed between the PD and the control. And the motivation for this was double. On one hand, maybe we will find some circRNAs implicated in the disease, although I agree that we are looking at almost at the end point of the disease. This is post-mortem samples, people who have been very sick. So you could never know whether this is consequence of the disease, consequence of even the treatment, or has anything to do with the disease. But on the other hand, we also wanted to find ways to biomark Parkinson. And of course, you cannot biomark from brain samples, but potentially you could extend this approach to other type of samples. And we found this particular circRNA generated from a channel which is SLC8A1. And this channel is expressed widely in the brain, even in cell culture, but was only differentially expressed in the substantia nigra of the PD individuals. And here you can just take a look at the data. So the circRNA is upregulated in the PD patients, but we don't see much difference on the mRNA. So the main difference is on the circRNA levels, not on the mRNA levels, suggesting that either the circularization is altered on the PD patients, or something else like the circRNA degradation maybe, but it's not increasing the amount of mRNA from the gene. If you try to correlate the amount of mRNA with the amount of circRNA, we see a strong correlation in the control individuals. So the amount of circRNA that you generate from the locus is directly related to the mRNA. Okay, this suggests there is some co-regulation of the two molecules. And we wanted to see whether this co-regulation is, because we saw no change in the mRNA and change in the circle, we suspected that there is this balance between the circle and the mRNA is lost in the PD patients, and this is indeed the case. So the PD patients actually have lower amount of the circRNA with no change of the mRNA, so this balance between the circle and the linear RNA is lost. In parallel, to be honest, the PhD student working on this project was trying to see whether a particular circRNAs get induced when we treat cells with oxidative stress agent like paraquat. And More tried this in a few different systems, and all of them were quite consistent. And what we saw actually was that almost the only circRNA that will increase the level following paraquat exposure in a dose-dependent manner was this circRNA. So by two independent ways, by looking at the brain of PD patients or by treating cells with paraquat, we found this circRNA going up dramatically with oxidative stress. Okay. Again, I would say that the mRNA didn't go up. So this strongly suggests that what is happening is that upon oxidative stress, the production of this circRNA is dramatically increased. And remember, circRNAs are very stable molecules. So having 2.5-fold increase after 24 hours treatment is dramatic, because the levels of the circle already are high. So to increase it even further, you really need to increase the production. Moreover, we also treat the cells with the statins or with the LRK2 inhibitors, and we saw the circRNA dramatically going down. I need to say these treatments also make the mRNA and the protein to go down. But again, I want you to realize that the circRNAs are very stable molecules. So almost the only way to go down is to be degraded. Shutting down transcription alone is not going to make them go down very dramatically in a short time. So this suggests that there is a specific regulation of the circRNA, which is increased by oxidative stress and is decreased by factors that act as neuroprotection. So the question is, how does the circRNA work? And we don't have a complete answer for this, but I think we have a good hint of what the circRNA could be doing. So as I told you before, circRNAs originally have been described as molecules that can bind to microRNAs and regulate the function of the microRNAs, or the localization of the microRNAs, or the stability of the microRNAs. But other circRNAs have been implicated in other type of molecular functions like binding to proteins, or translation. So we try to see whether any of the known models of action of circRNA could fit this data. And we actually look in clip data, and we found that the exon that makes the circRNA, which is strongly conserved between human and mouse, has been found to have sites for microRNAs inside, and actually to have clip reads. This means when people have done AGO-RNA interaction, and they look for a sequence of RNA that bound to the microRNA machinery, they have found those sites which are conserved between mouse and human. So this means in that particular exon, the second exon of SLC8 in human and mouse, this RNA is bound to the microRNA effector machinery. And it's very unlikely that this is to regulate the mRNA, because most mRNAs are regulated at the 3' end. And you can see also peaks here, suggesting that this mRNA also bound to AGO, but in the 3' end, like usually mRNAs are. So in order to see whether this could be the case, or what hypothesis then that maybe the circRNA was bound to AGO, the first thing was to check that the circRNA is actually cytoplasmic. And as you can see, the circRNA is strongly cytoplasmic, not nuclear. So it produces the nucleus, but then export it. We also perform an AGO-IP. So AGO2 is a protein that links microRNA function to mRNA decay. And what we did, we just basically did a biochemical assay. We IP the protein, and we look at the RNA to see whether the circRNA is bound. And the circRNA is strongly bound to AGO2. Strongly suggesting that those clip reads found in human and mouse, they are due to the binding of the circle, not the linear RNA. When we try to see which microRNAs are predicted to be bound to this circRNA, we found 7 microRNA from miR128. So miR128 is a microRNA which is strongly expressed in the brain and has been linked before to neurodegeneration. So we took it as a normal candidate to mediate this effect. And we try to see whether this circRNA could mediate any of the effects. So in order to do so, we try to see whether when we have in the brains of the individual with PD or in cells treated with paraquat, we can see a change in the levels of the target genes of the circRNA when the microRNA, when the circRNA is being increased. No, I just mixing up a little here. So remember in the PD brains or in the paraquat treat neurons, we see an increase of the circular RNA. And together with this increase on the circRNA, we wonder what happened to the targets which are relevant for neurodegeneration. These are some of the miR128 targets. And we see that both of them go upstream, so are upregulated, suggesting that at least these two things are correlated. And I need to remind you that the initial hypothesis is that circRNAs are important for sponging out or degrading the microRNA, but actually the molecular data show the opposite. The circRNA seems to be important for stabilizing the microRNA. So in this case, looks to us when the circRNAs go up, the microRNA seems to be going down because the target go up. Okay, so sorry if I was not clear, but I don't feel this shows any causal relationship, but it's just a correlation showing that the two molecules at least are co-regulated. And we are studying this further in vitro to try to understand exactly what the circle is doing to the microRNA, at least in cell culture. So this leads us to a model, and the conclusion from this part of the study is that we have identified a few circRNAs which are differentially expressed from 3 different brain regions in control and PD, individuals with PD. This is post-mortem. We identified this particular circRNA as a circRNA potentially involved in PD, at least misregulated in PD, and that could be used to biomark potentially the disease because this gene is highly expressed in blood, for example. We are extending this approach to blood, hoping to biomark PD. And for this, we are still analyzing the data that was collected in Israel. And we are also analyzing a large data set generated by the MJ Fox Foundation that has a lot of data on circRNA because it's total RNA sec from total blood samples. And the blood sample we have from Israel are monocytes. So I want to take the last few minutes, now I have still a few minutes, to tell you a little more what we know about circRNAs in aging, and which is potentially relevant for PD from other projects in the lab. So this will be very fast, just to show you. So in my lab works mainly in flies. So most of the work we do in the lab is in flies, although we also work in mammalian cell culture, as you can see before, and samples, human samples. But we basically, flies have a lot of circRNAs, many of which are conserved between flies and mammals. And what we did is we generated huge resources, genetic resources, in which we can knock down specific circRNAs one by one and look at different parameters. So in particular, we look at young and old flies, and we did all sorts of behavioral parameters as well as gene expression. Again, we really characterize around 115 lines in which we knock down specific circRNAs. So we found circRNAs important for many processes. Here, some of them are important for development. Here, just showing the viability. Some of them important for development. Some of them are important for locomotor activity. So when we knock down the circRNA, we have flies which are hyperactive. They move more during the day, or they move less during the day. Either some of them are important for sleep and for circadian rhythms. But the ones that we found, and we have a lot of behavioral phenotype associated with those knockdown of those circRNA, in particular synaptic defects, which we are trying to understand. And this mirrors a little what people have found when doing this in mammalian systems. Brain development, especially synaptic development, synaptic maintenance. But the most exciting result we got, I think, is that most circRNAs don't affect lifespan, but we have found a few circRNAs that can actually extend the lifespan when we knock them down. Some of them, they decrease the lifespan, and this could be just because these circRNAs are very important for regulating feeding, for example, or for just regulate the normal function of locomotor motor neurons in one of the cases. But we were really excited to see this, that we have 4 or 5 circRNAs that when we knock them down, specifically increase the lifespan, more the health span than the lifespan. And this result is very consistent and very strong for these few circRNAs, and suggests that maybe as the brain ages, the accumulation of the circRNAs and the potential, for example, translation of these molecules could be a burden for particular cells within the brain that they make the aging process to be faster. We have verified, these are just 2 examples, the 2 controls and the 2 different SH lines targeting the circRNA. They dramatically increase the lifespan in this for these 2 particular circRNAs. We did RNA sequencing, and sorry, this is a very crowded slide, it's also a new slide. We did RNA sequencing on all these flies. So we have flies which are very young, 3 days old, 15 days old, and 30 days old. And we did RNA sequencing. And this is a principal component analysis in which you just basically put the whole data and you see how the data is similar or different to each other. And what I want you to focus is here. So these are these three flies, as you can see, either the control flies, these are the control flies, or the other ones are the knockdown flies. They all group together, the young flies all group together. But if you see 15-day flies are going to group around here, and 30-day flies are going to group around here, okay? So what I want you to see is that the control flies in every group are going to be more mixed with the older flies. These are the control flies for 30 days old, while the flies in which we knock down the Circarians that we believe are involved in aging look like younger flies than the control. And this make us think that the problem of these flies, or the advantage of these flies that they live longer when we knock down the Circarian, has to do with the flies being younger from not only the dying point of view, but also transcriptome point of view. And we have done now several behavioral assays that strongly make us believe that the Circarians in the brain may be a burden, at least these particular Circarians during aging. So we have found a few Circarians that extend the lifespan, and we have found that they have a younger transcriptome. So this effect is in aging and not with some acute effect. The next step, we are trying to identify the pathway that led to the work of these Circarians. And we have now a lot of genetic tools to use this and to investigate this further. So that I really want to thank the people that did this work. The work has been done, most of the work has been done by More. More was a PhD student share between my lab and the lab of Mona Sorek at the Hebrew University. And other people in my lab contribute to this work, but this was really a partnership for the last few years with Mona and the people in her lab. And I forgot to say, Eres Lebanon also collaborated with us, help us very much with the editing analysis, and very grateful to the MJ Fox Foundation for support our research on this topic across the years. Okay, that's all.
Sebastian, thank you. That was wonderful. First of all, a very good and thorough introduction to the field. I think for many of us, it's really very new. We needed that. But then the transition into how you were able to apply that to Parkinson's and aging is really, really fantastic. So one thing I'm intrigued about is you said that you've had the funding from the Michael J. Fox Foundation for quite some time. So it seems that they really were interested in funding this in the very early days of when you were doing this work, when probably it was still a hypothesis in the involvement.
Yeah, and I think that the main motivation from the MJ Fox Foundation was to try to see whether we can use them to biomark the disease. And the first step was to try to see whether they have a function in the brain. And now we actually, some of the, we have been mining their that database, and there is clearly, I'm confident, I'm positive, I would say optimistic that we will be able to find a few Circarians that are going to be differentially expressed in individuals with PD, even maybe in the early stages. So that's something that we look forward, and yeah, it would never have been done without their support.
Very rewarding. Wonderful. I think that's a very positive signal to funding agencies, is give the funding early on when it's really critical, not wait till there's already a whole grant worth of preliminary data and then only then fund it. Okay, so let's open up for questions. Nir, are you there to MC the questions? Why don't you, please?
Yes, thank you. Thank you really for a very stimulating topic and a wonderful overview. So it's open for questions. Maybe I can start with how variable is this from subject to subjects?
Yeah, that's actually a very good question, and I need to say that the samples, our data set was deep but not crazy deep, and we couldn't see this variability. But in the PPMI data, we see that we have around, when we look at 3,000–4,000 subjects. So you have at least I'm talking about in blood, no? You have a few thousand Circarians which are expressing all the individuals. but which is a good number, no? A few thousand, 2,000 maybe, which are in almost all the individuals, but you have tens of thousands or hundreds of thousands of circRNAs which are expressed in only one or two. So the question is what this tell you. I believe that tell you that there is a lot of noise on the circRNA expression because many of them are very lowly expressed. But you have a core of circRNAs which very highly and consistently expressed across individuals.
Questions?
Hi, Sebastian. Nice to see you. I joined a bit late, so maybe I missed part of what you said. Have you been looking for changes between gender that are known to affect Parkinsonian patients?
Yeah, actually, the decrease of circRNAs is stronger, especially in the effects are stronger in males, which a little fits the clinical data. But again, we are looking in individuals with PD already. So this could be just a consequence of the disease rather than the cause. But we didn't, I need to say that we didn't see big difference in the age accumulation with the male and the female. I mean, there was a few things that we were looking even in the control patient, but we didn't see. But we saw a more dramatic effect on the males, which again, you cannot tell whether these are postmortem samples, so you cannot tell whether this is a consequence or has any causality, but it's a good point. I need to say that from the data from MHA Fox, we have imaging, we have like tons of parameters. So, and they have been following this cohort for a few years and they intend to follow. So I think from that particular data, we will be able to correlate a lot of parameters observing the disease with the number of levels of the circRNAs.
Okay.
Hello, Sebastian.
Hi.
Thank you for your talk. I think you said that the circular RNA could be a burden on the cell, if I'm not wrong. Why is it?
Yeah, so this was very surprising for us. So this was observed for some circRNAs. We see that when we knock them down, this is in flies, but the same result has been now reported, or I have heard it is also happening in other systems. So they accumulate dramatically with age. We're talking about molecules which are very abundant, and maybe some neurons are more sensitive to this accumulation than others. So these circRNAs, for example, can recruit ribosome for translation. And maybe imagine if they are in a particular, I mean, to make the answer short, we don't know why. Okay, we just see this, that when we knock them down, we extend lifespan. Those particular circRNAs we overexpress and we shorten lifespan. So they seem to be a problem having these circRNAs in the cell, but the circRNA accumulate like crazy with age. Really, you have a 5-fold accumulation, these particular ones even more. So then you have this molecule that maybe can bind to ribosomes, maybe can be translated and make a toxic peptide. We don't know if it's because of the peptide. We don't know if because association with particular proteins or not, but we are studying. So for what, it's not every circRNA, most circRNA, when we knock them down, we have no effect on lifespan, no extension of lifespan. But there is a few circRNAs that we see an extension.
So they could be a byproduct of gene editing or something like this?
It's not a genetic effect because this is control. The genetic background is control, and we are just knocking it down. So we have control for the genetic background. It's not the genetic background, but probably something, some gain of function that happens only with age. Because if you look at these flies when they are young, they look very similar to the controls, but the difference only appears when the flies are middle-aged or older.
Nir, I have a question.
Sure, go ahead.
So going back way to the beginning when you were describing the RNA-seq and then the identification of the circRNAs. So is this kind of in the RNA-seq that I have of different tissues, the circRNAs are the data is there, it's just that I haven't processed it properly to be able to find them? Is there certain preparations you have to do in order to see them? It's a certain skill of the bioinformatics?
Yeah, that's a great question. So So basically, it depends how you did RNA sequencing. A lot of RNA sequencing is done by mRNA. You basically purify mRNA with oligo-DT or oligo-DT bits, and then you do a library. Other way to do a library is just to deplete the ribosomal, just to sequence everything. You deplete the ribosomal RNA and you do a library. So circRNAs, you need to do ribosomal RNA depletion, because if you do poly-A selection, you're not going to see that. And then you use a specific pipeline, and we actually developed a new one. It came out the paper a few months ago that to identify. But the preparation is, I mean, many people prepare the RNA by ribo depletion, so maybe your libraries are ribo depleted. I don't know.
What is the offside, and what's the downside of doing that if you want to get, if you're doing the regular standard RNA-seq?
Yeah, I mean, to be honest, the price of making a library with poly-A or ribo depletion is almost practically the same, but it just is more usual. You know, people usually do it by RNA purification, mRNA purification, but depends. I would say if 50-50. We have mined a lot of data sets, and I believe the ribosomal depletion is better because you get all the information like splicing, pre-mRNA, you know. So, but depends on the facility or depends how you generally do RNA-seq.
I just wanted to ask, beyond doing a knockout, is there any other way of degrading these circular RNAs?
Yeah, so the only physiological way to degrade a circRNA, at least known in nature, is basically by shRNA. So if you have a small RNA, which is fully complementary to the circle, you're going to degrade it. Of course, in our case, we target the shRNA to the junction. And in nature, again, there is two examples of a circRNAs which are degraded by this method. But in the two cases known for this, the two circRNAs are produced from loci which don't make any mRNA. So the microRNA in this case, which is fully complementary, is going to degrade the circle, but there is no effect on the linear because there is no linear from that locus. And those are very special circRNAs. But I'm pretty sure that circRNAs are degraded in vivo probably by other pathways, because some of them, there is no complementary to any known small RNA. And we know their half-life is very short. Again, if you inhibit, in certain preparations, if you inhibit neuronal activity, then you see circRNAs completely degraded together. And upon viral infection, circRNAs are known to be degraded by RNase L, but RNase L is not, for the best I know, to be active in synapses. So probably in the brain, there may be other molecules, other RNases that could degrade circular RNA, but they are unknown. That's a good question.
Thank you.
Nir, before you close off, if I could just make one comment. Some of you may have noticed I changed my background, so I'm doing a little bit of free advertising here. We're organizing a healthy aging conference. It's going to be in person at the Museum of Natural History next Wednesday and Thursday. And if you don't find the information on the website, please send me an email and I'll be happy to send you the registration details. So all of the Israeli speakers will be giving the lectures live on site, and we have a lot of goodies, good food there. And our British guests, because this is the BIREX UK Israel, are actually going to be Zooming in. Everyone will be able to listen to it either in person, and you could also register and then get a Zoom link if you want to do it in the privacy of your own office or home. So I hope that many of you will join. And also to let you know that we're starting now a Center for Aging at Tel Aviv University. We still haven't figured out the name. We're still working on that. So if anybody has any ideas that they'd like to contribute, we'd welcome them. We're just about to start. We got some funding to do so. And we'll certainly see, even in this lecture already, that there's so many common areas, both in Parkinson's disease and aging. And so I think that there'll be a lot of collaborations that can happen between the scientists and the clinicians who are involved in both centers. Thank you, Nir.
As a follow-up, I'm going to talk about the aging effect in Parkinson's disease in that symposium. So it's all linked together. And thank you, Sebastian, for sharing this exciting observations that you have been doing over the past almost a decade now. And it's really a new area that might have a significant impact on our understanding of aging, of Parkinson, and the linkage between the two. So thank you everybody for joining us, and we will have the next talk next month by Professor Roy Alkalai, who just moved back to Israel and will share with us his view on Parkinson's disease and biomarkers. So we'll see you next month.
Okay. Thank you very much.
Thank you. Thank you.
Thank you.
Thank you.
Thank you,
Bye
Karen.
bye.
Bye.
Bye.